carries through this water column into the xylem and all the way down the roots. So in a way, it's like sucking water up through a straw. It's creating a tension, a negative pressure at the surface of the leaf that's transmitted all the way down to the roots of the plant through this water column in the xylem. And this pull from the negative pressure generated by this transpiration, cohesion of the water molecules, and this overall tension moves the water from the soil to the root, into the roots, and up to the leaves. And this doesn't require any energy on the part of the plant. It's strictly a physical process. All right, so again, here we see this basically, we have the lowest water potential, or the lowest pressure potential at the atmospheric level here, and we have increasing pressure potential as we work our way down through a vein and then down through the xylem to the roots and then to the soil, which has the highest water potential in this system. So in this case, the lowest water potential again here, the highest water potential here, a gradient of decreasing water potential, and water moves up with a strong cohesion of the water molecules together and the adhesion of the water molecules to the cell walls of those vessel elements and tracheids. So that water column is continuous, it's unbroken, and it moves up just due to that physical process. All right, so of course, it's really important for the plant to regulate these stomata to keep from losing too much water. And the way the plant's, basically the way that the size of this pore is regulated is through these guard cells that flank it. There are 2 guard cells, one on each side of the pore. And each one of these has cell walls that are thicker toward the side facing the opening than on the other side, as you can see here in this tan-colored region, thicker here than here. And the way the microfibrils in the cell walls are oriented, as the cell becomes increasingly turgid, as water enters it, as it expands, it bows outward. It bows out, can't expand as much along this side as along this side, and that opens the pore. Okay, so that's the general way. When it's not as turgid, then it closes. And so the way that plants open this, we'll talk about it. Actually, they have photosensors that can detect whether light's present or not. Light is a stimulus to opening these stomata. There's no point in having them open if it's dark out. Photosynthesis is impossible. You're just going to lose water unnecessarily to the atmosphere. We'll talk about photoreceptors in a little bit. But light is perceived, and then a proton pump is activated. This is active transport now, moving protons out of the guard cells. Of course, protons have a positive charge. And because of the movement of protons out, we're going to get a charge along this cell membrane of the guard cell, positive to the outside, negative to the inside. And that's going to allow then for potassium ions, which have a positive charge, to more readily move into these cells and out. And that concentration of cations inside this guard cell will then result in a higher solute concentration inside the guard cell than outside, and water then is freer to move in than out. Through osmosis, it moves in, and we get turgidity of these guard cells, and they bow outward, and the stomate opens. And then, for example, under dark conditions, this whole system reverses. The potassium moves out, and the water moves out following it through osmosis, and it closes. All right, so the stomatal regulation is incredibly precise. Plants, a little bit of cloud cover comes over, stomata will start to close. The plant really regulates those, pretty fine-tuned. But under a drought condition, for an extended period of time, plants are really up against it if they can't bring enough water into the plant to make up for water loss. And even with the stomates closed, plants can start to lose water just through the cuticle. And eventually you get what's called cavitation, where under sufficiently negative water potentials, under sufficiently negative pressure, and I should emphasize here that this type of water flow through the xylem is the pressure component of water potential completely makes insignificant the solute concentration differences in this water column, and strictly a pressure-driven system as far as the xylem goes. But the negative pressure potentials that can build up inside of these xylary elements, in some cases can cause the xylary elements to collapse altogether, but before that happens, usually we have an air pocket forming. And once we get an air bubble forming inside the water column, that breaks the cohesion of the water molecules, and the transport ceases. So that tension is removed, and we don't get further movement. But remember that those xylary elements have pits along their margins, and tracheids and vessels are not just in one pipe. There are many of these tracheids and vessels alongside one another, and if the column breaks in one of these xylary elements, water can move out the sides of a xylary element that's beneath that point into an adjacent xylary element, and the column can maintain its integrity. You can still have water movement. But once we get down to a certain point, we get too many bubbles forming across this region, and eventually the whole water transport's impossible. And then the plant's in big trouble. And it can't necessarily recover. A little bit of cavitation, and the plant can recover at night if it can rehydrate to a certain level. The air bubble can dissolve back into the solution. But beyond a certain point, it's over. And horticulturists take advantage of this with sophisticated systems for actually keeping track of the water potential inside the xylem and making sure that watering occurs before it gets too low. All right, so flow and transport, that's it for xylary transport. Now we're going to go to how the other type of vascular tissue transports sap. And of course, in the case of phloem, that sap is not water and dissolved inorganic minerals, but it's photosynthate. It's basically sugar water. It's syrup. It has the consistency of syrup. It's like 30+% sugar. And of course, that photosynthate is being produced in the leaves, and it has to get, for example, to the roots to nourish the roots. So the transport's occurring in the opposite direction of the water transport in many cases. And so this type of mechanism we're talking about doesn't work, the kind of mechanism that happens in xylem. So the transport of phloem sap is from the source where the photosynthate is being produced, the sugar is being produced, or where it's being mobilized. In the case, for example, of a storage organ, like a storage root, in the springtime, starch is being converted to soluble sugar, and that would be the source from where that sugar would then be moving to the growing parts of the plant. But in any case, it's going to move from the source to the sink, and the sink is just anywhere where that photosynthate is being stored or used at a higher rate than it's being produced, if it's being produced there. So in a typical plant in the middle of the growing season, the source, shown in green here throughout, is the leaves that are fully developed, and the sink, shown in purple, are going to be leaves that are still growing, needing more photosynthate than they're producing, and also, for example, in inflorescence, it's not producing photosynthate, but it's using a lot of energy, and the roots, which are also needing to respire, they're using energy, but they're not producing any energy like photosynthesis. All right, so thinking about how phloem sap moves, basically the model for phloem sap is what's called the pressure flow model, and the sugar solution, the phloem sap, as I mentioned, is going to move from the source to the sink, and the source is an area of higher sugar concentration, the sink's an area of lower sugar concentration. And basically, phloem can be modeled with this simple little device shown here. We have a closed tube, a U-shaped tube that's full of solution. Sugar solution is higher on the left side of the tube than on the right side. And the tips of this tube are immersed into pure water on either side, in bit flasks of pure water with a semi-permeable membrane that separates the ends of these tubes from the pure water that allows water to enter freely or go out freely and doesn't allow the sugar solution to move into the flasks. And as I'll point out in a minute, this is a pretty good proxy of what's happening in the plant. And so water, by osmosis, is going to have a higher tendency to move in than to move out because the sugar is binding up the water inside this tube. Water's freer on the outside here. Through osmosis, it's moving in more than it's moving out. And it's also moving in on this side faster than it's moving in on this side because there's a much less concentrated sugar solution on this side of the tube. And so basically, just because more water is moving in here at a higher rate than it's moving in here, we're going to have net movement in this direction, and we have pressure, positive pressure, pushing water from the left side to the right side. And that's the pressure flow model. And that's what we would call an example of bulk flow, just as it is in xylem, where we're moving from a region of higher water potential to lower water potential. So we look inside of a plant. This is a longitudinal section of the vascular tissue. Here's the phloem over on this side and the xylem over on this side on the left. So the plant's going to be loading sugar into this phloem sieve tube. And in flowering plants, at least, this is an active transport process where we're concentrating sugar inside this phloem sieve tube to a higher concentration than in these adjacent cells, the companion cell that, remember, is paired up with the individual sieve tube element of in phloem, the sister cell. So active transport is resulting in concentration of sugar here. And then at the sink end, we have sugar being actively transported out into a sink cell out of the sieve tube. And these processes together, up at this end, moving the sugar molecules into the phloem, is going to result in, through osmosis, water is going to move from this relatively pure water solution in the xylem into the phloem sieve tube. And at this end, when we have the sugar being pulled out of the sieve tube element, water is going to be moving back out into the xylem by osmosis. And so we have positive pressure developing at the source end and much higher pressure here than at this end. And bulk flow is then going to be in this direction toward the sink. And so again, this is a type of bulk flow where we have higher pressure at one end than the other. And that's how, basically, in angiosperms at least, that the phloem moves through the phloem sap moves through the phloem. So I'm thinking about the phloem sap movement. The sinks are usually relatively close to the sources. So in the case of a growing tip of a shoot here, it's going to be deriving its photosynthate from the upper leaves of the plant. In the case of the roots, that sink there, it's going to be drawing most heavily on sources that are closest in these lower leaves here. So that's another thing to consider, that it's that the movement of photosynthate is relatively proximal in different parts of the plant, from the closest source to the closest sink. All right, so in terms of comparing xylem transport and phloem transport, they're similar in the sense that these are both examples of bulk flow that's due to differences in pressure between different parts of the plant. And in this case, it's basically less negative pressure inside the root than in the leaves, but the pressure still is higher down lower in the plant than up at the top. In the case of phloem, it's also a pressure difference, but it's positive pressure at one end relative to the other. And so, whereas in this case, it's basically more negative towards the leaves. So these are both good examples of bulk flow. Simple diffusion couldn't allow these substances to move in the plant as quickly as they need to. So any questions now about, yeah? Well, this would just come to equilibrium. There wouldn't be, beyond a certain point, there wouldn't be net movement of sugar in the phloem. Yeah. So the process doesn't require light to operate, though. I mean, depending on whether the plant, the plant could continue to do this after dark. There's no requirement for light for this process to occur. Does that make sense? Yeah. As long as photosynthate is available, that is. I mean, that's the limiting factor here, as long as there's soluble sugars that are available for transport. All right, so we're going to move on to how plants actually sense light. So they don't have a nervous system. They can't look up in the sky and say, oh yeah, it's light out. We can see the light. But they do have photoreceptors. And these photoreceptors are very sensitive. They can not only tell whether it's light out, they can tell the direction of the light, how intense it is, and what the quality of the light is. And the way that they do that is they have a couple different types of photoreceptors. They have blue light receptors that are perceiving light down in this part of the spectrum. And they also have, and basically these blue light receptors allow the plant to orient to light. They're important in orientation to light. They also are important in the functioning of the guard cells. Remember, I mentioned how the guard cells can perceive light, and that involves the blue light receptors down this end of the spectrum, invisible light. And then there are also phytochromes, which are photoreceptors that are operating in the red and far red ends of the light spectrum. So these photoreceptors are interchangeable. So there's a form of phytochrome that's called red and a form of phytochrome called far red, and they're interchangeable forms of phytochrome. And the relative balance of those two forms of phytochrome allows the plant to have a very nuanced understanding of the intensity and quality of light. And that's important in seed germination in some plants. It's important to shade avoidance in the way the plant grows, out from under shade, for example. And also is important in the plant's ability to detect day length and whether it's light out or dark out in general. And in order to think about the importance of day length in physiology, first we need to think about circadian rhythms, which apply to all organisms, including us. These are endogenous rhythms of an organism that can be reset by external stimuli like day length, but to some extent are physiologically dictated. And if you've ever had jet lag before, if you've flown to Europe or somewhere across several time zones, you know that it doesn't just take us 1 day to get readjusted. The body takes a while for its internal rhythms to be reset based on external stimuli. And these body rhythms include a lot of different factors in physiology. And this is true for plants too. If you put them in total darkness, they'll lose the ability to, they have rhythms, but they'll run at a longer day length, about 26 hours rather than 24 hours. And pretty soon they're pretty far out of whack until they get back into a normal light regime, and then they can reset. So in terms of being able to detect and respond to light, one of the most important types of responses in plants is photoperiodism, which is basically physiological responses to the relative length of night and day. And an important photoperiodic response is triggering of flowering in plants. Some plants are completely day length neutral. They'll flower whenever they get to the point where they have enough resources to do so, regardless of the day length. But a lot of plants will not flower until the day length is at a certain level. And of course, day length changes through the year. And photoreceptors such as phytochrome are playing a really important role in this day length response. And there are two types of plants that have two types of responses. There are short day plants and long day plants of those that have photoperiodic response of flowering. And unfortunately, these terms are not completely accurate because physiologists have subsequently found, it was found that plants would go to flower in some cases when day length increased beyond a certain, or decreased or increased beyond a certain level. And in other cases that plants would flower when day length dropped below a certain level. But it turns out that that's actually a response, a measuring of the night length as opposed to the day length. And short day plants are plants that flower when night length increases past a critical threshold. And long day plants are plants that flower when night length decreases past a critical threshold. And next time we'll talk about how that actually works and how it's experimentally shown before we move on to the final bit about plant growth responses involving hormones and signaling.